Hello and welcome everyone to the fourth public science panel brought to you by the University of Washington's astrobiology program. Thank you for joining us live on YouTube. And a reminder that you can follow the UW Astrobiology channel, which is full of lectures and videos all about astrobiology. You can also find some of our previous public science panels with topics like the best place to look for life in the universe and the origin and nature of life itself. So my name is Trent Thomas. I'll be moderating the panel today, and I'm a PhD student in the University of Washington astrobiology program. Tonight, I am joined by four of my fellow scientists who are all driven by their curiosity about the possibility of life in the universe. So, is there life on Mars? It's a question that has been at the forefront of humanity's collective mind since the red planet was discovered. From the hypothesized Martian canals proposed by Percival Lowell in the 1890s, to the 1971 hit song Life on Mars by David Bowie, many great minds have contemplated this question. So tonight, we will meet four astrobiologists who, like Lowell and Bowie, are investigating whether or not there is life on Mars. Our panelists are experts in different aspects of this multidisciplinary scientific problem, and they will share their insights in a moderated discussion for the first part of the panel, and then they will answer your questions after that. And you can type any of your questions in the YouTube chat box. I'll also advise you to pay close attention to what the panelists say tonight, because there'll be a fun and friendly competition later where you can showcase what you learned. More on that later. So without further ado, let's begin by getting to know our panelists. So, panelists, please say a few words about who you are, what you study about Mars, and if you could bring one thing to a Mars colony for entertainment, what would it be? We'll start with Elena.
Hey, Trent. Okay, thank you so much for inviting me. This is really fantastic to be here. So my name is Elena Amador French, and let's see, I have a PhD from the University of Washington's astrobiology program in Earth and Space Sciences and Astrobiology, and my focus was on orbital remote sensing of the surface composition of Mars. Essentially, from a global perspective, what is the surface made out of compositionally, mineralogically, and then how do you place that into astrobiological context? So like, are there certain minerals that indicate certain environments that we learn would be very conducive to life? And so I spent a lot of time digging through lots and lots of data, scouring different regions of Mars to think about, could these different places have had habitats essentially for early Martian putative life? And so a lot of my work in my postdoc was working on candidate landing site selection for Mars 2020. That was very relevant and cool. And now I work at NASA's Jet Propulsion Laboratory, and I'm a science operations coordinator for the Curiosity rover. And so with my science expertise, I am kind of the scientists in the room at JPL who interface daily with the engineers here, who are the ones, sequencing the rover's arm movements or drives, and making sure that the engineers and the scientists are speaking the same language, which can be very difficult sometimes. So it's really fun to be that bridge and to learn both worlds. Okay, if I could bring one thing to a Mars colony, it's cheesy, I'd bring my daughter, my 2-year-old toddler, because she's very entertaining.
That is a definitely respectable answer. We'll just have to make some room in the colony, I guess. So next, Joan Chen, how about you go?
So I'm Joan Chen. I'm a PhD student at the UW Astrobiology program and also atmospheric sciences. So I'm interested in the chemistry of trace gases and airborne particles on Mars. Currently, I'm studying how the surface of some airborne particle can catalyze some of the chemistry happening on Mars, in particular the productions of perchlorate from chlorine-containing gases. So I'm using model to study this kind of chemistry problems, and understanding this kind of chemistry can help us to understand and also to constrain how the Martian atmosphere and soil will behave and evolve in the long time scale, and also the likelihood for finding chemical fossil on Mars. From the entertainment to bring to Mars, I guess I will bring a beer brewing kit with me to Mars and try to make some craft IPA on Mars. Yeah, trying to bring some life to Mars. Yeah.
The first Martian brewery. I'm sure you'll have a big market.
Yeah.
So great. Yeah, perchlorates. More on that later for sure. So how about Andrew? You're up next.
Hi, everyone. I'm Andrew Shumway. I'm a PhD student at the University of Washington in the Earth and Space Sciences Department, and also in the astrobiology program. In my research, I study water on Mars, because if there's any life on Mars, it's going to need water to survive. I can't go to Mars myself to study that. So instead, I try to recreate a little piece of Mars in the lab. I think of it as like having Mars in a jar. And in that jar, I can control the temperature, the humidity, and other environmental factors to study what happens to water under those conditions. And so if I was going to Mars, I would bring probably my guitar, because otherwise Mars would be a very quiet and sad place.
I would love to hear what rock formations inspire your music on Mars. It'd probably be very beautiful. So last but not least, Ken, would you like to go?
Yes. So hi, I'm Ken Williford. And I'll also just say thanks so much for the invitation. It's always so good to be able to do anything in connection with UW astrobiology, having been a product of it myself, the program. And it was just such an important part, such a crucial part of my education and of my life generally. I feel really fortunate to have been a part of it. And I left with a PhD in late 2007. While I was there, I studied a mass extinction event at the Triassic-Jurassic boundary, which is about 200 million years ago, focusing on organic and stable isotope geochemistry. So extracting organic matter from rocks deposited across that mass extinction event and trying to understand the interactions between the conditions that led to the mass extinction, trying to understand what caused it, and the response of the biosphere, and in particular, the recovery of the biosphere. From there, I did a couple postdocs, one in Australia at Curtin University, in Western Australia, where the best, oldest record of life on Earth is preserved, which was one of the draws there. And I started working on progressively older rocks at that point. And then I spent a couple of years in Madison, Wisconsin, in a lab there using a technique called secondary ion mass spectrometry, which perfectly combined my interests in microscopy and organic and isotope geochemistry. SIMS allows us to, rather than having to grind up a rock and measure its chemical composition to get the information out of it, we can slice it and take beautiful microscopic images of it and do spectroscopy at a much finer scale than what Elena was talking about, but some of the same techniques. But then shoot the targets that we're interested in analyzing with a beam of cesium ions about, say, a tenth of the diameter of your hair. And so we would call those kind of techniques spatially resolved analysis. And it turns out that's a big theme for Mars 2020, the mission I currently work on as deputy project scientist. So I help lead the science team for that mission. And then I started a lab at JPL called the Geochemistry Lab. So those are my two main jobs at JPL. But around Thanksgiving time, my family and I moved back to Seattle, where we've wanted to settle for a long time. And so I actually work remotely, and I'm probably down the road a piece from a lot of the folks on this call. So.
Awesome. And if you had one thing you could bring to a Mars colony for entertainment?
I forgot that part. Yeah. So I first, like Andrew, thought, well, it's my acoustic guitar. If I ever have a choice to bring one thing anywhere, it's that. And then Elena said her daughter, and I thought, I'm a horrible father, so I should bring my 4-year-old daughter. So because Andrew is bringing his guitar, I will also bring my daughter, and Elena's daughter and Dorothy can grow up on Mars together. That sounds good.
I hear the schools are great on Mars, so I think you're taking them to the right place. Well, great. Nice to meet you all. Thank you for joining us tonight and introducing yourselves. And I'm sure I, just like the audience, look forward to hear what you all have to say about our favorite planet, the one right behind me. So with that in mind, we are here to talk about our next door neighbor, Mars. But in our solar system, there are plenty of other planets and plenty of other moons to go around. of all these, Mars gets all the attention. Well, most of the attention, I'd say. And I mean, look no further than right here. We have an entire panel dedicated to Mars tonight, and it was even voted best place to look for life in a previous panel from the series. So this question is for all of you. What exactly makes Mars such a tantalizing place to look for life? Why does it get all the attention?
I can take the first start at this. I think there's several reasons. There's the scientific reasons why it's really interesting to go look at Mars. So we know that life on Earth started, Ken could give the exact time period, but 4.2 billion years ago or so, and there's evidence of those rocks in Western Australia, for example. But there aren't very many of those rocks left on Earth because the rock record on Earth has been mostly eroded away because we've got this pesky weather system and atmosphere and plate tectonics and all these things that just destroy all our rocks. And so Mars, fortunately, we know from mineralogical evidence and other modeling that early Mars probably had a thick atmosphere, at least episodically so, and there was water flowing around that same time, about 4.2 billion years ago. We have evidence for lakes and rivers, etc. And then on top of it, the rocks are preserved. So there's, this whole southern hemisphere of Mars is these ancient, what we call Noachian, 4 billion year old rocks. And because there's no plate tectonics the way Earth has it, and there hasn't been this long sustained atmospheric hydrological system, these rocks are preserved and available for us to go investigate. And so this is really tantalizing because we know that at this time period on Earth, life was forming. There were probably similar environments and habitats on Mars at the same time, and those rocks are accessible. So that's like one of the scientific rationales that makes it really tantalizing. And then there's like the political, practical reason. It's really close. It's rather close. We know how to get there. We can get there in about 9 months with a spacecraft. We've got teams of experts at JPL and around the world really that know how to do the engineering. And so we've learned how to do that from an engineering standpoint. So like these two factors put together make it a really great place to send your rover if you only have, 1 or 2 spacecraft to send in the solar system to go look for evidence of life. That's my first jab at it.
Yeah, no, those are truly compelling reasons. Does anybody else have anything else they'd like to add to that?
Yes, I'll say one quick thing. It's probably as good a time as any to make the distinction between looking for evidence of ancient life versus looking for evidence of extant life, or life that's currently living or recently deceased. And I certainly couldn't have said it better than Elena did. Those were exactly the 2 reasons I was thinking about. I would say in a sense, those are both largely focused on Mars's great potential to preserve this extremely ancient record of early planet formation and maybe even the time of the emergence of life on a planet, which, as she said, has been wiped clean on Earth. But the key thing is, and life, maybe I'm getting ahead of ourselves, but life could be on Mars today. But if it's there, we think it's almost certainly restricted to the deep subsurface, or at least I'll put my opinion out there that that's the case. And so Mars astrobiology is generally, so far and for the foreseeable future, until we can drill very deeply on Mars, it's restricted to this surface record of very ancient rocks. Whereas if you want to go looking for evidence of life, as most people understand that, can we find living creatures elsewhere in the solar system? It might be easier to do it on some of the ocean worlds, where we know there are currently habitable environments. Now there are a whole other set of challenges about actually getting to those habitable environments on the other places as well, but we can maybe talk about that later.
Yeah, absolutely. Well, that's great. So as Elena and Ken have alluded to, there's a lot of evidence that there was water flowing on the surface of ancient Mars. And certainly that's what some of the purpose of the rovers being sent up there is, to investigate that even further. So that said, ancient Mars may have been warm and wet, but today, as you can see behind me and Andrew and Joan Chen, it is cold and dusty. So I have to wonder, where did all of that water go? Is any of it still on the surface or the subsurface? So this question is aimed at Andrew. Yeah, where did the water go?
Yeah, thanks for that. You're right. There used to be like rivers and lakes on Mars, but they're long gone by today. Like you were saying in the photos behind us, you can see it's just basically looks like a dry desert with not a drop of water in sight. And the reason has to do with Mars's magnetic field. On Earth, we have a strong magnetic field that basically creates like a shield around the Earth that protects us from the sun's most harmful radiation. So here on the surface of Earth, we're blocked from most of the most dangerous particles that come off of the sun. But on Mars, there's not a strong magnetic field like that. So the worst of the sun's radiation is able to just hit the atmosphere and the surface of Mars. What that means is without that magnetic shield to protect it, over millions of years, the sun has stripped away Mars's atmosphere. So today, Mars has barely any atmosphere at all. And because of that, it's really cold and dry. It's so cold that on a warm summer day, you'd be lucky to get above like freezing temperature. So any water that might be left on Mars would be frozen year round or would evaporate into the air. So there is still a way to find liquid water on the surface. If you think about snowstorms here on Earth, for example, the roads might freeze solid. But how do we then melt that ice and clear those roads? We use salt, right? We salt the roads. That's because salt lowers the temperature that water freezes at. And just like our snowy roads, the surface of Mars is covered with salts that can melt that ice and form salty waters, also known as brines.
Great, brines. Yeah, the first thing that comes to mind on the subject of brines is pickles, for me at least. Can anything actually live in a Martian brine, or is it just kind of there for show?
Yeah, it definitely depends. We need to know how salty the water is, because all living beings need water to survive, from us humans to the tiniest microbes. Like Trent, you and I, we need fresh water to keep on living. If we drank salt water, though, we would get sick, we'd end up in the hospital, it would be just all around not great. But a fish, on the other hand, a fish is perfectly fine swimming around in salt water, drinking it. So all life collectively has some limit for how salty of water it can use to sustain itself. So maybe you've heard of the Dead Sea here on Earth. It's one of the saltiest bodies of water. It's called the Dead Sea because it's so salty that almost nothing can live in it. Fish, plants, nothing can survive, except even if you scooped up the water from the Dead Sea and put it under a microscope and looked at it, you'd see little fungi, little bacteria swimming around. So even in one of the saltiest bodies on Earth, you could still find life. So the big open question on Mars is, how salty is that water? Is it salty enough where life could maybe be floating around in it? Or is it just way too salty for life altogether?
Fascinating question, fascinating. I'm glad you're working on it. I'm glad many people are. And so, yeah, the water is fascinating. The chemistry of the water is equally fascinating. But another equally interesting thing is the soil chemistry and the atmospheric chemistry on Mars. So this one is aimed at Joan Chen. Is there anything that makes Martian soil or the Martian atmosphere particularly suitable or unsuitable for life?
Yeah, so just like what Andrew said, the soil on Mars is also heavily influenced by the evaporations of brines. So it contains a relatively high level of salt. And on Earth, a lot of terrestrial organisms, such as trees or vegetables, do not like salty soils. And one more interesting features about Martian soil is that it contains quite a high level of perchlorate, and this is a pretty surprising findings from the Phoenix missions. And perchlorate itself is in the context of Earth is a environmental pollutant from our industrial activities, and perchlorate can interfere humans' hormone system. So the perchlorate containing Martian soil is likely quite toxic to humans. But although like this kind of Martian soil is quite toxic or unsuitable for complex life to thrive, some microbes actually like them. So on Earth, microbes, there are a class of microorganisms called halophiles can thrive in extremely salty conditions like the Dead Seas or like the soil in Atacama Desert or even blind pocket within the sea ice. And then in addition, some microbes on Earth can actually use perchlorate to carry out respirations under anoxic conditions. So if we put them on Mars, they may actually make use of the perchlorate in Martian soils and grow happily from them.
Yeah, fascinating. Life finds a way, huh? Even with toxic perchlorates all around them.
Yeah.
So far we've gotten a wonderful cross section of microbes where they could live on Mars, from the salty brines, feasting on nasty chemicals, or perhaps the ancient warm and wet conditions where it seemed like anything could live there. But throughout all of these different environments and throughout all of Mars's history, there has been intense change. I mean, it's been a theme we've been talking about pretty much throughout this discussion so far. So my question now to all of you is, how could life originate and survive in the face of all of these intense changes? I mean, is one microbe suited to go through every environment, or do they kind of hunker down maybe? So anyone take a stab at that one?
One thing your question brings to mind for me is a scenario that we think about with Mars 2020. Our mission is the first leg of a sample return campaign of several missions. So whereas Mars 2020, one of our main jobs is to seek evidence of ancient life with the instruments on the rover, we're also using those instruments to select samples that we think have a high likelihood of preserving any signs of ancient life that was there in the Jezero Lake environment. But say we're totally successful and we find some tantalizing biosignatures, signs of life with the rover instruments, and we take a core of that, we get it back to Earth, and we say, hey, eureka, we found evidence of ancient life on Mars. Of course, the next question is, does that represent what's sometimes called a second genesis, or a second origin, an emergence of life independent from one that happened on Earth? Or is this the result of panspermia, or of exchange of living organisms between Earth and Mars? Say life originated on Mars maybe, and a giant impact threw up a rock containing some of those Martian microbes. They landed on Earth 4 billion years ago and seeded our planet, and we're all ancestors of we're all Martians in a sense. Or maybe it happened the other way around. Or life didn't originate on either of these planets, and it originated in around some other star and made its way here. And so this concept of panspermia is something that's absolutely a viable scientific concept as far as we know. And so that's the big question that discovery of evidence, of clear evidence of ancient life on Mars would raise, because there are very different implications, I think, in terms of the basic question of astrobiology. One of the basic questions, how common is life in the universe? How commonly does life emerge from non-life? We don't know how it does. There's a bunch of models for how it might happen, but also how special is it?
Yeah, great points. Does anyone else want to jump on top of that, say anything else?
Yeah, I'll just add really quick. I mean, I think one of the key things that Ken just said is, we don't know how life originated on Earth. Like, that's still an open question. So trying to speculate about how life might originate on Mars it's a very broad question. One of the things I worked on during my PhD was studying rocks that form in serpentinizing environments, which some folks think perhaps these are seafloor rocks that interact with water. There's olivine is a mineral and it interacts with water and it releases hydrogen. And some folks think that perhaps the origin of life may have happened on Earth in these subsea floor environments. That's one hypothesis. And so one of the things I worked on in my PhD was looking for evidence of this mineral serpentine that has very distinct spectral signatures on Mars, well, on Earth, and looking for them in data of the surface of Mars. And that was just one, and we found that evidence in certain places, including in the watershed of Jezero Crater up in Northeast Syrtis in the Nili Fosse area. So that's very tantalizing. Like there are minerals that form on Earth in environments that we think life may have originated in. It's one hypothesis. And those same minerals were forming on Mars at that same time period. And so that's super cool to think, okay, there could have been serpentinizing environments on Mars 4.2 billion years ago as well. And so those rocks are very interesting.
Yeah, bring up the idea, using what we know about Earth to try to make inferences about Mars. So great. So I want to kind of segue now to our robotic exploration of Mars to give the audience a taste of, what we've done and what we have learned from these exploration missions. So this one is directed to Elena and Ken, our people at JPL. So what do you think are some of our most important astrobiological quests to Mars? There's been a lot, I know.
Well, often when I talk about Mars 2020 in sort of broad settings and try to paint what we're doing in broad strokes, I will sometimes say that Mars 2020 is the first mission since Viking that really has an explicit objective to seek signs of life. That doesn't mean it's the only mission since Viking that had capabilities to seek signs of life. Absolutely. MSL, Elena said she was doing MSL operations earlier today. The Curiosity rover has fantastic capabilities to find signs of life. But there has been this sort of progressive evolution of the sort of the mode of the search within NASA that started with this concept called follow the water. And so and this is all post-Viking. So the reason I brought up Viking was this is at this heady time, the Carl Sagan Cosmos days in the mid-70s. He sent this lander to Mars just to go and reach in the soil and put some soil in the biology experiments and ask the question, does life currently exist on Mars? It was a very different time. We knew a lot less about Mars than we know today. Then there was almost this dark period when the results were fairly negative. Really, I would say the consensus is that there was no evidence for life on Mars from Viking. But interestingly, Gil Levin, the PI of one of those experiments, recently died, and he maintained for the rest of his life that his experiment found evidence of life on Mars. But since, after that, the rover era started, and the early approach was to follow the water as the kind of unifying feature of all known life. If you can find geologic evidence of water and confirm that, where at the time there were raging debates over whether these features we observed on the surface in images that looked like flowing liquid, whether that was evidence of flowing water or lava, some kind of volcanic processes. And the early rovers confirmed that indeed there was liquid water on Mars. And then Curiosity took that to the next level and said was about looking for evidence of a habitable environment with a lot more subtlety and nuance than just simply was there water there. And then Mars 2020 takes that another step further and builds on all of those approaches and very directly looks for signs of that ancient life using spatially resolved analyses and some of these really pioneering new instrument techniques.
Yeah, great. So let's, I want to take an opportunity to kind of hone in on Curiosity and Perseverance. So I want to start with, could you tell us a little bit about what techniques Curiosity is using to investigate Mars and what they have shown us so far in the time that it's been up there?
Yeah, so Ken did a nice job of talking a little bit about Curiosity's objectives and goals. So Curiosity was never sent to Mars to look for evidence of life. The objective was to answer the question, was Mars ever habitable? And so, yeah, for the longest time, we had this follow the water. We thought that water was it was one of the key features that we thought we needed for life. But as we've learned more about what life on Earth needs, we've learned, okay, it's more than just water. It's an energy source. It's, climate conditions. It's, what's the other one that I'm missing? Nutrients. I'm trying to think of my diagram of what life needs. So there's more to life than just water. So Curiosity's instrument suite was really put together to say, okay, was Mars habitable? And we sent Curiosity to an ancient lake bed, Gale Crater, where it looks like there may be some inlet channels, but really it looked like there's twofold. One, it's a crater that has sediment in it, but the sediment, very interestingly, compositionally transitions from clay minerals. So clay minerals on Earth form with the interaction of water and rock, and they have water bound within their mineral structure. And then the minerals transition from clay, and then as you move forward in time, so to more recent time, they go to sulfate rich rocks. And so the rock suite, the sediment suite that the Curiosity rover has been investigating for almost a decade now, is this really interesting snapshot in time in Mars, where you go from, maybe 3.5 billion years ago to more recent Hesperian, early Hesperian rocks. And you transition from what minerals that imply a wet, thick atmosphere to a more arid, acid-driven environment. And so it's this cool snapshot in time. And I think that's really the key thing that Curiosity has shown, is that, yes, there was water, but there were also redox. There are minerals that imply redox gradients that we know life needs to harness energy, to metabolize. There are chinops, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So these elements that are really the bedrock of life. And so Curiosity was able to kind of check that box. That was, kind of the first goal. And then now we're looking, now that we've said, okay, yes, Mars was once habitable, we're getting into the more nitty gritty of, okay, so how did the environment change over time? Was it just like always wet and then it was dry? Or maybe like we were wet and then we were dry, and then we were wet and we were dry. And like there are multiple episodes of this. And there's like really interesting veins that run through the clay rich rocks that imply like episodes of like fracturing the rocks and filling them with sulfates. And so that's been really interesting. And then the other big thing that Curiosity has is the SAM instrument suite, which has been, not only is it looking for like organics, but we can do these atmospheric measurements. And so we've been observing methane over the course of the mission in the atmosphere, and learning that there's like this diurnal cycle of methane in the atmosphere at very low concentrations. And so that's been very cool. But Curiosity never had the goal or the objective of finding life. We have a hand lens. We have like a little hand lens that a geologist would carry that, if there was evidence, some structural evidence, if we saw the fossil bone, or, if we saw a stromatolite, we would be able to identify it with the instruments we have. But that's kind of the level of life detection that we have.
Yeah, interesting stuff. It was, we've learned a lot from Curiosity, thank goodness. And so Curiosity's, I guess, younger sister, Perseverance, is up there roving around at the moment. So Ken, I guess the same question to you now for Perseverance. What is Perseverance looking for, and how is it going so far, I guess?
Yeah, we, Elena mentioned participating in the landing site selection effort. So we had this 4 or 5 year effort prior to launch in 2020 of Mars 2020 in the Perseverance rover, where we got any scientist who was interested in weighing in on that to help us whittle down from something like 40 or 50 potential landing sites representing all different kinds of geologic environments. We had a lot of lake type environments, and then some other really interesting ones that were focused on some of the kind of chemistry that Elena mentioned with serpentine and the interaction of this mineral olivine, which turns out to be very primordial. It's sort of the first silicate mineral, the first thing that looks like a crystal that crystallizes and precipitates out in the solar system before the planets form. And Hawaii is made of it. Almost all of Mars seems to be made of it. And when you react that mineral with water, all kinds of interesting stuff happens. Clays form, hydrogen can be generated as a fuel for microbes, et cetera. So environments, even if there's no lake there, but it's just rocks that look like they have olivine in them or similar minerals, and you can tell they've interacted with water, those are very interesting places to look for life in maybe a form that's very different from life as we know it on Earth, or put it a different way, to use a different kind of exploration approach, whereas almost everything we know about the history of life on Earth comes from fossils in shallow water environments. So think coral reefs, or the shallow edges of a lake, things like that. That's where fossils form best. So we know almost everything about life's history on Earth from those kinds of environments. It's also true that life, as deeply as we can drill on Earth, when you bring up a rock from kilometers below the surface, you find life in it, in fractures in the rock, even if it's an igneous rock that formed at very high temperatures. Once it fractures and water gets in there, microbes make their way in there, and they use the chemical reactions with the water and the minerals to liberate these energy sources for their metabolism, and can live quite well down there. So we looked at other landing sites like Northeast Syrtis that I think was mentioned earlier, that's just outside of Jezero to the southwest, as a place to look at the ancient subsurface record. And this is kind of a new frontier in astrobiology. Look deep, look in the subsurface. In the end, what we chose was a compromise where we would land in Jezero, land in what is very clearly an ancient lake environment. And we already have that mapped out. I mean, it's just so beautifully exposed. We have the rim of the crater. We know where the boundaries of the lake would have been. So we know where those shallow water environments would have been. The delta, the deep water environments in the lake. We have the river flowing in, the river flowing out. But then this terrain outside. So if we're so fortunate to do our exploration all around Jezero and still have a healthy rover, we can get outside the crater and go down toward Northeast Syrtis, toward that different kind of ancient subsurface environment, maybe even much older rocks or somewhat older rocks than we find in Jezero, and open up a whole new realm of geologic diversity, all in the interest of both looking for evidence of ancient life on Mars, understanding the earliest record of planet formation that, like we've said before, on Earth, that whole record is completely destroyed. Any rocks that are older than about 3.5 billion years old are generally so altered by metamorphism and plate tectonics that it's hard to read much in them. Whereas on Mars, things are beautifully preserved back to 4 billion years old, we think. And so we'll look while we're there, we'll take samples, about 30 or 40 samples, and then some future missions will come and grab those samples, put them into Martian orbit, launch them actually off the surface of Mars in a rocket, like a bowling ball size thing with 30 samples or so in there. And then an orbiter will be in place to grab those samples, bring them back to Earth, where that opens up the entire scientific arsenal of planet Earth to work on those samples and really try to find, use all the best techniques we can to confirm whether Mars was ever inhabited. And just the last short thing I'll say is our payload, our set of scientific instruments was designed with the kind of work that I do and my other colleagues who work on the earliest record of life on Earth. We tend to focus on using microscopes and these sort of spatially resolved analyses. So rather than grinding up the rock and losing all that contextual information, I still make lots of measurements like that, where we grind up a rock and make a bulk measurement. But it's so powerful when you can scan a beam across something that you've taken a beautiful picture of, because the main thing we're looking for are lifelike shapes. Elena mentioned stromatolites, these layered rocks that we think are fossil microbial mats, lifelike shapes with lifelike compositions. So things that have carbon, the schnapps that Elena mentioned, and different metabolic ingredients, and especially where they occur together, where you get lifelike compositions and lifelike shapes occurring together. That's how our payload was designed. And so it's a lot of potential to make some great discoveries.
Yeah, spatially resolved analysis, shining lasers at rocks, always fun. And so with all that in mind, just one more question before we move to the audience Q&A. So we talked about sending samples back to Earth to hopefully see if life is in those samples. But something sometimes people don't worry about as much is, what if we're sending samples to Mars? On these spacecrafts, Are we contaminating Mars with, microbes that have like clung to the side and then get off on Mars? And it's a really fun taxi, I guess, for 9 months. So this question is for Andrew and Yuk Chun. What is planetary protection and why should we all care about it?
Yeah, I think you put it really well, Trent. Planetary protection is about protecting Mars from Earth life, and also, for the flip side of that, is protecting Earth from any life we might bring back from Mars, like Ken was talking about bringing those samples back. At this point in the pandemic, we're all really familiar with this idea of accidentally spreading germs without noticing. So planetary protection is about making sure that that doesn't happen on a planetary scale. We don't want our rovers to accidentally bring germs to Mars that might act like an invasive species and contaminate the environment there, and maybe even kill off native Martian life. That's a hypothetical. So we have to be extremely careful to clean and sterilize the spacecraft before sending them up there. But even when we try to clean and sterilize, you can only get so clean, right? Like even the best hand sanitizer works like 99.99% effective, but that still means that some germs are left behind. And they obviously do a lot more work to sterilize the rovers that we send to Mars, but there's always going to be something left behind. So you got to be really careful not to bring those rovers into environments where those germs that are left behind might hop off and maybe contaminate Mars.
Yeah, so maybe one more thing to add is that right now, because of the planetary protections policy, I think the missions also have to avoid sites that are known to have maybe liquid water or water ice near the surface to avoid contaminating Earth life to the Martian environment. So right now, we are also avoiding the places that are most likely to for finding Martian life because of this policy.
Yeah, something definitely important to consider that sometimes, slips your mind. You don't think about it. We so want to find life from Mars that we gotta, restrain ourselves and make sure that we're not the ones putting life there in the first place. So now I'm going to start reading off some audience questions. But before that, I want to introduce the interactive component for the audience tonight. So we've talked a lot about the potentially habitable environments on Mars and the ways in which we've explored them. But now I will pose the question to you, the audience. What do you want to know about Mars and how do you want to investigate it? Tonight, I'm asking you to devise your own hypothetical instrument to go on the next Mars rover. And of course, it needs an outstanding name and perhaps a really good acronym. So let me share my screen here. Where is it? Everyone can see that. Submit your hypothetical instruments at the link here, tinyurl.com/ab-instrument. And bonus points will be given for using things you learned tonight, entertainment value and creativity, and for the best acronyms. So at the end of the panel, I'll read off some of our favorites. So without further ado, let's start off the audience questions by asking the question that is the title of this panel. Given everything that we've discussed so far, how would you, the panelists, answer the question, is there life on Mars?
I'll take maybe the cautious answer, which is it is too early to tell. It's maybe the more boring answer, don't have a hot take for you, but definitely we've explored so much of Mars, but there's so much that we haven't explored. Like Ken was mentioning the deep underground of Mars, we've only really gotten into the very, very surface or the very top few centimeters of Mars. There could be lava tubes where there are these caves that life could survive. There are all these fantastical scenarios you can think of, like life living in lakes under the ice in glaciers in the North Pole. Many, many different ideas have been proposed, and we have so much more research to do and exploration to do to answer that question.
Yeah, good answer. Anyone else want to add to that?
I'm going to give the eye-rolly answer, which I think there's two parts to it. One is, as Andrew was talking about, we probably have sent Earth life to Mars at this point on our spacecraft. It's very likely, and tardigrades will live through anything, like radiation. I am I have a good feeling there's at least one tardigrade on one of our rovers. And so that's one answer. And then the other is really cheesy, but like we are all on Mars every day. Like you can log in and see images that are like downlinked directly from Mars every morning, you the public. And so I mean that's the cheesy answer, is that we are there. Like we've sent our tears, sweat, blood, you know, our souls to Mars.
Yeah, great. It's a tough question for sure. And anyone else, any burning opinions to say about it, or else I'll move on to an audience question.
I already said mine, but I'll say it again. Maybe in the subsurface, in the deep subsurface.
Yeah, Yak-chan, do you want to add anything, since everyone's going?
Yeah, so I agree. Like we probably already brought some life to Mars already by sending some spacecraft. But like the more interesting answer to this question is that any Mars originated life on Mars currently, or like in the ancient history of Mars, and currently remain as some fossils that could be discoverable by us.
Yeah.
And maybe I'll just add one other thing about ancient life. Is the answer is super interesting either way. Like if we discover there's no life on, like if we're able to definitively, which I don't know if we'd ever be able to say, but like either answer is super interesting from an astrobiological planetary evolution perspective of whether there is or isn't life on Mars.
Yeah, great point. It's fascinating either way. So I'll move on to some audience questions now. So starting with a question by Jim K. So related to the Martian atmosphere, what is your theory regarding the possible origin of methane detected in Mars's atmosphere? So if someone wants to take a stab at that.
I mean, I mentioned the methane, so I'll talk a little bit about it. The reason, so methane was detected, gosh, I'm not going to remember the paper. I want to say in like 2010-ish time period, there was methane detected in the atmosphere of Mars from Earth-based observatories. And it was a big deal because, as a lot of us know, methane, the major producer of methane on Earth are microbes, cow farts, if you will. And so for the longest time, people thought like, okay, if you find methane, perhaps that's evidence for a extant, like a present day source of microbes exhaling methane. But there's also abiotic ways of producing methane that are very ancient, that we were talking about the serpentinizing reaction. So as we said, hydrogen is one of the components that happens from serpentinization, but that hydrogen can then react in Fischer-Tropsch type reactions with carbon dioxide and create methane. So it's an abiotic source of methane. So perhaps, and I think what is most likely the case, is that the methane that we've been observing is just methane from either past water rock interactions on Mars that have been trapped in the rocks and are being released. There's this really interesting diurnal cycle that we've found with Curiosity. And so you could kind of think like, okay, maybe as the rocks contract and expand in the day-night cycle, perhaps you're like releasing a little bit of methane. The other interesting thing they're finding is there's like a, in the atmosphere, like an elevation, an altitude component to that. Like Curiosity will detect it at a different time or different concentrations than like the overhead, like the trace gas orbiter. We've done some correlated observations. So I think it's likely that they're just water rock interactions that are creating methane that we're observing at very low concentrations.
Yeah, it certainly made big waves in the articles and all the news that came out around it. So yeah, great points. So another question from Jim K. Again, good questions, Jim. What are the panelists' perspective on the Martian meteorite AH84001, so Allen Hills meteorite, regarding the interpretation of its evidence of possible fossilized life from Mars? If anyone wants to have a go at that.
I'll have a go. So I don't take the features observed in ALH 84001 as evidence of fossil life on Mars. I don't think we currently have any evidence, compelling evidence of life outside of Earth. So the field is wide open, in my view. But at least in kind of the circles we run in, the Alan Hills story, that work often gets a bad rap, I think, improperly. I think that it's sometimes used as an example because of all the hype and excitement that came out after that paper in 1996. Bill Clinton giving a press conference in the Rose Garden, and it kind of went from there to, I would say, the broad view is that there is not evidence of ancient life in that meteorite, but there's so much interesting stuff in there that tells us a lot about the history of Mars. And the way that paper was put together, and I encourage anyone who hasn't looked at it, whether you're a scientist or not, to go dig it up or email one of us if you can't find it. But dig it up and look at it. The approach that they took, it really is very similar to the approach we take today. It combines imaging and geochemistry, spatially resolved analysis. Often a lot of attention is given to the shapes in there, and it was, I think, overly simplified as well. We found some interesting weird looking shapes, so those are fossils. It clearly was not as simple as that, and the authors really did a much more nuanced job than that. And then the last thing I'll say is it's sort of like the paper that launched 1,000 ships. I don't think I would have a PhD. In fact, I'll go as far as to say, I don't think we would be having this panel today if not for that paper. The NASA Astrobiology Institute came right on the heels of that paper, and all the attention that came into the field of what was previously exobiology and generated, really, in a sense, this new field, at least within NASA, of astrobiology, and the funds that paid for the earliest astrobiology work at the University of Washington, right around the beginnings of the field of astrobiology, and then this educational component that led to such a great educational program at UW. So it's just an incredible piece of science and a scientific story that then beyond the astrobiology aspect, we've learned so much from the mineralogy and some of the isotope work done on the carbonates and other phases in that meteorite. So it's a great rock.
Yeah, the rock that launched 1,000 ships. Awesome. Yeah. So another question to the audience now from Charveri Capri. Sorry if I mispronounced that. But if we ever do get to study the brine water on Mars, how likely is it that there would be extremophile-like organisms like halophiles? And how do you think they would differ from halophiles on Earth?
I can take that one. So yeah, the main difference is, I'll start by saying that it is likely that if we did find life in the brines, they would be halophiles, which basically means just salt-loving. The halophiles on Earth are only able, like I was saying earlier, to withstand a certain level of saltiness, though. And the thing is, the salts on Mars are much, much more saltier than what we're used to finding on Earth. So life on Earth can only withstand so much. So if there was life on Mars, my guess would be that they would have to withstand a lot of salt, like way beyond what we're seeing any life being capable of here on Earth. So it could be that is just not possible, and that once you reach that limit, there's no life that can survive, which is what we see here on Earth. But it's also possible that that's just because that's how life on Earth has evolved, right? If there's life on Mars, maybe it's evolved and adapted to that Mars environment. And if there's life in the brines, they could then possibly survive those extreme saltiness. But the best evidence right now is that the salty water on Mars is not likely habitable.
Anything else from anyone else in the panel? If not, no problem. We've got plenty of questions to answer. So another one from, let me find it again, from Jim K, who is an AllStar Question asker, it seems. What would each panelist consider as a definitive indicator of ancient life and as a definitive indicator of present life on Mars? Tough question.
That's such a tough question. It's such a tough question that, Mike and I actually were just in a workshop where we're trying to come up with a standard framework for what would the community agree on would be a standard of evidence for ancient or extant life. It's so complicated because as you can see in this panel, we've got geologists, we've got chemists, we've got biologists. Like, that's the beauty of astrobiology, is it's so interdisciplinary. But what that also means that everybody has a slightly different definition of life. And so that can make it very difficult to come to a consensus. So I guess if we're going panelist by panelist, I think the obvious answer is, if you see, I think a fossil is a very easy, some shape that we can all agree on can only form from life. But again, you really have to make sure that there's not some mineral that can also make that shape. So it can be very complicated. I don't think I have a definite, maybe if you see something metabolizing with a heartbeat, but that's so earth-centered, right? So it's really hard. But it's something that we're all working on to come to agreement, because once we make the Eureka statement to the world, you want it to mean something. And so we have to make sure that we're all in agreement of what that will be, and what confidence, like how sure are we that this is? Are we like 10% sure? Are we going to, if you're 10% sure, are we going to make a statement, or we have to be 99.9? And how do you put a confidence on something? So these are very interesting open questions that the community is debating.
Yeah, definitely tough question. So yeah, let's just go around. Everyone should have given an answer to this, since it is so subjective, I suppose.
I liked what Elena was saying about actually being able to see it. So maybe if we, we're able to have a microscope and look and see things swimming around in the water, then that I would take that as pretty definitive proof. But maybe that's too high of a bar, because very likely once we get the sign that we will get, if we do detect life, will be nuanced. There will be some amount of confidence. How sure are we? So it is very complicated.
So I guess for me, I would look for a multitude of evidence. It's not just one image or just one isotope records, but just several of them combining together, and then giving us a measures of how certain that is a ancient fossils or a living life on Mars. And I think Mars is a great place for looking for this kind of multitude of evidence, because we have already studied it for a long time and observed its atmosphere, and also to learn about its geology for a long time. So we have already got the information of the environmental context of finding such a biosignatures. So combining the environmental context with maybe new information from the sample returned from Mars in 10 years, then we'll be able to answer these questions, or at least put a constraint on the certainties of finding life. Yeah.
I guess what I would add on to, I also like Elena's definition for ancient life, but we do actually, it's a very similar question to how do we agree as a scientific community on a set of features we find in the oldest sedimentary rocks on Earth? Are those indeed fossils or not? Are they biotic? It's called biogenicity evaluation, and we'll be doing the same thing. I hope we find some things with Mars 2020 that are interesting enough to warrant biogenicity evaluation like that. But if I had to guess, the most likely thing that we might find would be something like a stromatolite, a fossilized microbial mat. We are going into environments with Mars 2020 where if there were microbes in Jezero Lake, and if they were mat-forming microbes, which are common on Earth, they would be formed there, and they would preserve in those environments if they are if we have the mineralogy and the geology right. those environments. So I think it's absolutely possible. The added layer I would be looking for or hoping for would be isotopic offsets from two redox couples. That has come up earlier in our conversation. I would want to see some organic matter, so a carbon source, and I'd want to see some inorganic carbon that might be a proxy, a leftover mineral record of the inorganic carbon source for that microbe from which it built its organic matter. And I'd look at the isotopic offset in carbon 13 to carbon 12 between inorganic and organic carbon. We could similarly do that with other redox couples like sulfur. We could look at oxidized sulfur like gypsum, sulfate minerals, and co-occurring sulfide minerals like pyrite, where microbes might have transformed the oxidized species into the reduced species and set up a large isotopic offset. So those would be powerful biosignatures, as long as there are some biological looking shapes and these kind of strong chemical signatures occurring together, it would start to get really compelling. But where it would be most compelling is exactly like Elena said, a microfossil, a fossil that looks like a cell, and especially if there were multiple cells. And we have all these criteria. The cells should have a narrow distribution in size. They shouldn't be randomly distributed in size. And I could go on and on and on. In terms of modern life, it would be somewhat similar. I would want it to be made of organic matter, to have something like a cellular envelope, so to have an inside and an outside, and it keeps the inside different from the outside. And I'd want to observe metabolism happening. I'd want to see chemical transformations happening spontaneously from the outside to the inside, and of course, self-replication. And if it had those qualities, I would say that's alive.
And just one thing I want to poke at what you're saying, Ken, that I think is so important is, like, you're saying you need multiple lines of evidence. And with more lines of evidence, the more confident you are. And I think that's really key.
That's right. Yeah, there's no one smoking gun. We say that all the time. There are some shapes on Earth, as much as we like to say, the picture is never good enough. There are some shapes of stromatolites that we can't think of an abiotic way to form shapes that are this complex. But still, it comes with it. It has organic matter often, and you add these other elements in. So,
well, we always talk about the dinosaur bone. Like, if we see the dinosaur bone sticking out of the sediment, well, then But
yeah, we're not going to see a dinosaur bone. I will. I mean, never say never.
I think that would certainly surprise everyone if we found a dinosaur bone. But yeah, it's one thing we can all agree on is that it's a good thing that we're looking. So with that, that's the note I'm going to start wrapping up on, since it is about time. It's 7 o'clock Pacific. So before we go, and before I reveal all of the winners of our interactive component, hear their instruments, would you panelists like to tell the audience where they can find you on the internet, social media, or any way that you think that you'd like to engage with them? Start with anyone. Yeah, whoever wants to.
You can just Google my name and JPL, and you'll find me at JPL. And I'm happy to answer questions. So find my website and email me from there. I don't have other things, Twitter, or Facebook, or anything, but you can find me that way and send your questions.
Yeah, you can find me on Twitter at EAMADOR at Amador. And I often post about, Mars and operations, because that's kind of the world I'm in all the time, and also happy to answer any questions that anybody has.
Yeah, I'm on social media at Twitter at Humway, little play on words there. Yeah, you can also Google my name and find my page through the University of Washington Earth and Space Sciences.
Not an active user of social media, so you can find my email by Googling my name. So it's just ycchen@uw.edu.
Awesome. Yeah. And don't forget to follow the university.
One more quick thing, Trent. I'm also affiliated with this organization called Blue Marble Space Institute of Science. And I'll just take this chance to plug that organization, which really comes out of University of Washington, was started by some good friends of mine from grad school. And go check them out, and you can find me through there as well, but then find lots of other really interesting astrobiologists.
Awesome. Yeah. Thank you all for sharing. I hope the audience reaches out and engages in some real good scientific discussions. So don't forget to follow the University of Washington astrobiology page on Instagram. That is at uw.astrobiology. And then don't forget to check out the rest of our YouTube channel. We've got plenty of lectures and all of the past panels here to check out. And you can find me on Twitter at Trentagon with a 0. So Trentagon like Pentagon. And also a special thanks to fellow UW astrobiologists, Mike Wong, Lucas Pfeiffer, and Evan Davis for all of their incredible behind the scenes work. They make the show tick. And last but not least, let's reveal the winners from our instrument design competition. So I will share the screen once again. And here they are, right? Yeah, you can see this. Sorry, it's like my whole screen, but it's okay. So first one, Doug, the digger of underground germs from Rebecca Torsha. That's a great one. Next, the UWAB, maybe pandering. I mean, maybe it has a soft spot for all of us here. But the underwater atypical biology rover from Charveri Hemant Capri. I hope I pronounced it right. I'm sorry if I didn't. And finally, iCompromise, the complex protein measuring instrument. So exciting, from Billy Waits. All excellent instruments, and I'm sure they will be considered for the next rover that we send up there. Yeah, the last thing is make sure to come back and join us in September for our next panel. What is the future of humanity in space, with some more University of Washington astrobiologists. And this one will be moderated by Evan Davis. So please come join us for that. If you signed up for the last Eventbrite for this panel, yeah, you should get an email or be automatically enrolled for the next one. So with that, we will sign off. If everyone in the chat, give a big round of applause for all of our panelists. We thank them greatly for their time here and for their scientific expertise. So see you all in September.